Well, welcome everybody to this event that a number of us have planned to really talk about, investigate, question microbes and Alzheimer's disease, and actually some other diseases as well. And we think this is an important symposium to have to bring all of you together and all of us together that are working in this field to really try to understand how there's involvement of microbes and Alzheimer's disease and the importance of that involvement. I know many of you in the audience, the ones that I don't know, I'm Brian Balin. I work here at PCOM, the Philadelphia College of Osteopathic Medicine. I am a center director as well for the Center of Chronic Disorders of Aging, and I'm a professor of neuroscience and experimental neuropathology. And you'll hear from me too about the work we've been doing here at the institution. Nikki Schultek is here with us as well, and she has been a great organizer, the person who has actually been responsible for bridging the silos that we've been working in. And the bridging of these silos has required us to put away our egos and say, okay, let's work together, let's figure this out, let's move forward and make a difference in this world of Alzheimer's disease and dementias. Thank you, Nikki.
Thanks, Brian.
And so Nikki and I will both be initially talking about what we came to in organizing this. We want to first thank our sponsors, though, that are listed up here on the screen. It is wonderful to have sponsorship for an event like this. We feel that this demonstrates that there is an interest not only in our own scientific community, it is extended to our other communities, pharma, biotech arena, people that have an interest in funding this as well. Also, I want to, in general, just thank our funders that have a number of you being here. I want to make mention of Steve Sim and Marilyn Einstein, who have recently funded a number of our labs. So thank you very much. It is much appreciated to allow this work to continue. This is a slide I have up here also for this group. Kevin Zwezdarek, Amy Shapley, Michiko Thwe, Garth Ehrlich, Lavinia Alberi. This is a team that has put together this symposium and worked for many, many months on trying to get us organized to get this to happen. So I thank you very much for all your efforts that you've really put forth here. Of course, I need to thank the institution. I've been a really lucky individual to work at the Philadelphia College of Osteopathic Medicine. It's an institution where I came into from the allopathic world, and they wanted me to do my research, very supportive of that, to build research at the institution as well. And that's what we've done for me over the last 26 years. And I continue to do that and will continue to do that. So I want to thank our Center for Chronic Disorders of Aging, which is funded through the Osteopathic Heritage Foundation, which is a foundation out of Ohio that has been instrumental in funding different osteopathic groups, different institutions, to actually try to make a difference in the research world. And it was under the osteopathic symbol and basically approach, which fits perfectly with what we're talking about, because osteopathy is mind, body, and spirit. It really fits with what we try to accomplish here with our research, even though many of us are basic science researchers trying to understand what's happening with microbes and Alzheimer's disease. And then I have down here a list of names as well. And these names, I can't read all of them because there are quite a few, but these are the individuals that have supported us from anywhere from helping us get the communication out, to helping us with the laboratories, to helping us with just even at the front desk today, et cetera. And I value them because this wouldn't happen without them. I'm limited in my capacity to be that type of organizer, but we rely on all of these individuals. So I thank them. The symposium goals, what are we trying to accomplish today? And they're listed up here. In essence, this historical view for the last 30 plus years. An understanding of these microbes associated with dementia and Alzheimer's disease, this microbial dysbiosis, which is a big issue now with the gut microbiota, et cetera. And you're going to hear today from a number of people working in these arenas. The neuroimmune factors and host factors of how that plays in. And then some of the limitations and opportunities we have. How do we go about now developing this? How do we go into developing diagnostics and therapeutics that are going to really be effective here in this world? So that is going to be key. So now I'm going to introduce Amy Shapley, who is from the Infectious Disease Society of America, and they have a program that's been running for a number of years on microbial pathogenesis of Alzheimer's disease.
Okay, great. Now let's talk about well. So I just wanted to say thank you for being here. We're really excited to be a part and sponsor this program today. We do have some of our grantees that are speaking today, so I'm really excited about that. We've had a program for, we've had 5 cohorts of grantees. And so I'd love to put in your hands today 2 things. One is a book that has all of our grantee research outcomes to date. So I have a suitcase full of them. I don't want to take them home because they're very heavy. So if you would grab 1 or 2, that would be great if you want to see me during one of the breaks. And then we also have a Save the Part Symposium that's coming up, a Balin Symposium and a poster session in Los Angeles in October, on October 15th. And we're going to be hearing from some of our grantees, some of the people that are speaking today, as well as from other researchers. And then the last thing, again, if you want to come see me during the break, we are starting a program called Research Circles. That's really to try to increase collaboration between researchers in these areas and bridge more silos. This is an online platform that's researcher-led and researcher-created. So creating circles around the different factors that you might need to be shared resources. It could be sharing data. It could be working in a lab together. So come see me, please, at one of the breaks. I'll be up here watching time cards during the sessions, and I'd love to speak with you. Thank you.
Thanks, Amy. Is my mic working?
Yes.
Wonderful.
Now, Nikki Schultek will take it away.
Thanks, Brian. So I just want to say to everyone, first of all, thank you so much for being here today. I reiterate Brian's gratitude for everyone that it took to make this possible. Speakers, supporters, PCOM, of course, IDSA, all of our wonderful sponsors, and of course our AlzPI planning team, and then the whole AlzPI team. It is an absolute privilege to be here, and honestly, it's also a little bit surreal. 9 years ago, I never would have imagined I'd be standing in a lecture hall giving a talk on microbes in the brain or describing myself in the way that I will. So my titles are fine. What I actually do, I call collaboration architecture. So I bring people together. And what I bring people together around is the study of how chronic infections relate to chronic diseases, with emphasis on the brain and the lungs. But in order to really understand how I came to be in this sort of strange description of what I do, I have to kind of tell my story. And it did begin 9 years ago with being very sick. In my early 30s, I had actually just run my first half marathon. My kids were 3 and 5, and suddenly my well-controlled chronic lifelong asthma became severe and refractory to the point where I couldn't sleep at night. I also, over the course of a year, just began to deteriorate. I developed joint pain, a cardiac arrhythmia, a bladder pain disorder, which was one of the most painful things I've ever felt, brain fog, and the final assault at this terrible year were symptoms that were consistent with MS. It was, you know, a time in my life that I feel like I was robbed of a year. So when I see pictures like this, I think about how much I missed. So I have been fortunate enough to have a background after college. I studied business, not science, and then I worked for a big pharma company and then a biotech company. And during that time, I learned how to read and understand medical research, and I always loved science. So when I got really sick, I started accumulating lots and lots of specialists looking for answers. And I had, by the time I was, a year in, about a dozen specialists. Each specialist, what I noticed was they were focused on the one part of my body. So I would go in and I would say, hey, urologist, why bladder pain and my joints? I had this inflamed joint in my hand that was painful. Why my cardiac arrhythmia? Why that I stopped digesting my food properly? Why do I have neurologic symptoms? And everyone with a specific body part would say, look, I don't know, but let's talk about what I can give you for your bladder pain. So I noticed there was this lack of integration, right? And everything was siloed. So the picture you see here, so I've undergone at that time 4 chest x-rays, a chest CT scan. I've had in total 3 brain MRIs in my time since being sick. Countless other diagnostic tests, a VQ scan to rule out pulmonary emboli at the time because my breathing was so bad. And this photo was taken of me post-op, still woozy from anesthesia after a colonoscopy because I wasn't digesting properly. But I was fortunate enough to come across someone and something that effectively changed the rest of my life. So in my illness, I began to fear that if someone didn't identify the root cause of why I was systemically inflamed, that I was going to die. So I ended up doing a Google search that changed everything. I typed in interstitial cystitis and atypical pneumonia, 2 of my diagnoses during that time. And what popped up was my friend Chuck, Charles W. Stratton IV. He was the director of clinical microbiology for Vanderbilt for over 30 years. He was a board-certified internist as well as medical microbiologist and ID doctor. He actually wrote the board exam questions for ID and microbiology. He was a brilliant, brilliant man. So I came across a study he did with a urology colleague at Vanderbilt where they took urine from women with the condition I'd been diagnosed with, the interstitial cystitis, and tested it for an organism I'd never heard of, Chlamydia pneumoniae. So everyone knows typically chlamydia, the sexually transmitted infection, that's Chlamydia trachomatis. I didn't know about pneumoniae. I had no idea that this really prevalent organism floated around the human population. What the study showed, while it was small, too small to be powered to draw any huge conclusions, was that this organism that was detected in over 80% of the women's urine was capable of driving chronic inflammation because it hangs out inside our cells. And even though it's a respiratory tract infection, it travels throughout the body and immune cells. And at this point, my mind was blown. So I reached out to the corresponding author on this paper, Charles W. Stratton, Chuck. Never thought I'd get an answer. I literally wrote out my whole medical history and thought, he's going to think I'm insane. And within 5 minutes, I had a response. He said, get tested. And he sent Chlamydia pneumoniae, IgG, IgM. And the rest is kind of history. He ended up becoming my doctor. He ended up becoming my mentor when I started Intracell Research Group 7 years ago, unsure of exactly how I would help other than by bringing people together. And then he became my friend. And by the time he passed away 2 years ago, he was a part of my family. And you truly will never forget someone that brings you a torch in darkness. This man was about humanism in medicine. He was someone who truly cared about his patients. And I like to think that I was unique, but I wasn't the only one. There were other people who he showed his love and his gift to that had MS that he successfully was able to place into remission by treating infections. So what I got diagnosed with ultimately, I did have Chlamydia pneumoniae infection evidenced by direct detection actually that he coordinated through one of the academic labs actually. It's not even something that most people have access to. I do have Lyme disease. So Lyme is a bacterial infection that can mimic a lot of other things, but I think it's more than a mimic. I think these things are drivers. So I made a trip 3 months after Chuck initially did a telephone consult with my primary care doctor who was open to starting me on 2 antibiotics. My breathing immediately improved drastically. My condition improved. I will say that being treated for chronic infections is no walk in the park. There's a picture up there of me having a Herx reaction, Jarisch-Herxheimer. If you don't know what it is, look it up. But when a bunch of microorganisms like Borrelia die at once, or Chlamydia pneumoniae for that matter, a number of things happen inside the body and it sets off an inflammatory cascade. In very much the same way that patients being given chemotherapy may get sick before they get better, the same thing is true of Lyme patients. That photograph was taken of me and my husband and children outside the Ryman Auditorium the first time I went to fly and meet Chuck in person. Because after the 3 months of treatment, he indicated that my treatment likely needed to be longer. So that's the point at which he overtook my care. And I can honestly say that being heard is so huge for people like me. And I know there are other survivors out here in the audience that have been impacted by this, but gaslighting happens. You know, you go around to different doctors and still there's just a lack of education. And so that's one of our big goals here today, is to provide that insight. So what really blows my mind, this is to get into the science of it, but in a really basic way, we know that infections cause inflammation. Many chronic diseases have their roots in inflammation. You go to the National Library of Medicine and you look up inflammation, the first thing on the list is infection. But yet, why isn't it in the differential? So one of the big things that's hard with chronic diseases and infection is this notion of causality, right? So if something is associated with something, doesn't necessarily mean it's causing something. And I love that I call this my heavy seagull picture. But in this case, you know, we can use a picture like this to dismantle the discussion around microbes and Alzheimer's and say, well, you don't know that they're causative. They could be associated. True. However, what if the picture was somebody standing with a sledgehammer making contact with the fence? Do you still feel the same way? So I'm going to make the argument that microbes have the capabilities. They have the ability to get where they need to be. They have the ability, even from afar, to induce powerful immune responses, such as things that we observe with gut microbiome changes, as well as SARS-CoV-2, the COVID pandemic, you know, where something may not necessarily have to get in the brain to impact it. So this is just some of the evidence or categories of evidence that bring us all together today. Why are we all here about microbes and Alzheimer's? Well, they're found in postmortem AD brain tissues, so people that died with the disease, disproportionately, and different types of microbes than people who are healthy, age-matched, sex-matched controls. You also have evidence suggesting from one of our colleagues' labs here today, Dr. Will Eimer, and the late Dr. Robert Moyer, and Rudy Tanzi, Nandu Kumar, and others, that amyloid beta, which is the, quote, toxic substance that shows up in the brains of patients with Alzheimer's, actually might be an immune response or an antimicrobial peptide. You have the fact that animals that are inoculated with infectious organisms will go on to develop disease pathology in many different animal models that have been demonstrated. You have epidemiological data showing that people who get infections, almost of any kind, have an increased propensity later on in life to go on to develop dementia. And then one thing that is incredibly powerful that we identified in our work is an entity called reversible dementia. Now, I say we identified, we pulled together cases of infectious dementias and demonstrated that there are more than 80 in the medical literature, probably more that we miss, just depending on how you query it in PubMed, of dementias that patients presented with various forms of dementia, Alzheimer's, vascular dementia, frontotemporal dementia, others, and then were tested for infection, usually by lumbar puncture. Someone checked their CSF, not using any fancy technology, just using things that are available in a hospital microbiology lab, and found evidence of active infection, and then treated them with an agent and discovered that they could be, in the majority of cases, symptoms drastically improved. Some people were actually completely normal on an MMSE. That's compelling. And then we have the fact that there are other neurologic and psychiatric diseases that have been associated with or known to be caused by infectious agents. PANS and PANDAS that affect children, right? Neurologic conditions that are a response to strep infections. You have infectious neuroimmune issues across the human lifespan. So we have a new way of doing things here at AlzPI. We are really shaking things up by building diverse teams. It's a new model. It's an idea that shatters the concept that people that are Alzheimer's or neuroscience researchers should work on Alzheimer's. We have a team that is comprised of people like myself. I'm a patient. I am not formally trained in science. We have numerous specialties, and we encourage having debates, having meaningful discussions to make the most of what we can do together. And I'm so thankful to have a supportive spouse, my husband, Brendan, who's willing to creep around with me on construction sites to take pictures near silos. Thank you, Brendan. He's on Zoom. So, you know, like all things, they sometimes start out like a seed, you know, and then you grow them bigger. So that's the chicken scratch drawing of AlzPI, which was actually inspired by work by Dr. Michael Wilson at UCSF when I was reading about chronic meningitis and how we actually assess that. Dr. Wilson had a really cool small study where he tested patients with chronic meningitis, their CSF, using metagenomic next-gen sequencing, determined what microbial infections were there, found a diverse array of them actually, fungal, bacterial, parasitic, polymicrobial, viral, and then treated with targeted treatment to see if the patients got better, and they did. So I thought, oh my goodness, could we do that with Alzheimer's? Why not, right? So our team is really diverse. You'll see we have Dr. Brejwert, who's here today, who's a veterinarian. We have people working in biostatistics, data architecture and analysis, people working on fungus, people working on bacteria. We're intentionally diverse. And it turns out that this is not a bad idea. So this book is amazing. If you haven't read it, I highly recommend. But David Epstein asserts in a chapter called The Outsider Advantage that big innovation often happens from people that are educated, but they may be a little bit further from the problem such that they're able to reframe it. And in the book, he gives an incredible example of a situation at Eli Lilly with a gentleman named Alf Bingham, who started something, was basically like an innovation open source platform. And Lilly had these issues with some of their molecules that they were trying to resolve with this incredible internal team of chemists. And they cite this example where a patent attorney wrote into the open source platform and said, I have an idea based upon a tear gas patent I worked on. So literally solves the issue as an outsider. So this is what we're really trying to channel with our collaboration. And, you know, I have to say, the first thing we identified that was a major barrier to this space was the testing. So it was actually Dr. George Perry, who will be here later today, who pointed out, Nikki, how are you going to do this testing people and treating them, saying when the testing itself is inconsistent, right? We've seen studies come out on Alzheimer's and microbes. One lab finds something, one lab doesn't. Well, they're not using the same methods. There's no consensus. There's no collaboration. So we put together what we call this research roadmap, this establishment of a consensus protocol last year. It was published in Alzheimer's and Dementia, the Journal of the Alzheimer's Association. And for me, coming from where I've come from as a patient to co-lead that effort with Dr. Richard Lathe and my amazing colleagues that are mostly all here today, was a major celebration in the Schultek house. That was the day our paper got accepted. And I pulled out one of the reviewer's comments, which is hopefully this paper will catalyze the collaboration for finding a cure for this disastrous disease. One reviewer referred to it as transformative. And I'm really happy to say this is actually a real paper on PubMed. Take a hot second to read that. We thank the considerable physical distance separating our two universities, which prevented us from killing each other over minor details during the writing. So I will say everyone is marked safe after making the roadmap. You know, this huge team of scientists that came together. We did have some heated discussions. We wanted them, right? We want to debate things respectfully and disagree. You know, we can learn that way, right? We can avoid making mistakes. Everyone's healthy and safe. So this is a part of our paper in Alzheimer's and Dementia that I'm super passionate about. And it has to do with reversible dementias that I mentioned earlier. 86 cases. Just like Michael Wilson's work at UCSF with chronic meningitis, very diverse microbes. This is just a quick snapshot of the top of the paper. I encourage you to go through the cases. They're pretty intriguing. Cryptococcus neoformans, Borrelia burgdorferi, various viral infections, polymicrobial infections. But what this really tells us is that it should inform research. Cases are considered in some ways by the community, the research community, from what I've heard, a low form of research, right? It's an N of one case. Well, that's one person. And I'm a person standing here today, right? And others are here today that are well because of treatment targeting microbes. And there's a lot that we need to understand, but it doesn't mean we should shy away from it just because it's complex. So what are we doing here? So our primary goal at AlzPI was, one, to publish the roadmap. That's been done. Two, multi-laboratory detection. So we've enlisted some of the experts in the world doing different types of detection methods on the same samples so that we can develop what we refer to as the consensus detection protocol. Once we have this protocol, we want to use it in living patients and see what we find. Do we find evidence of ongoing active infection? Do they respond to tailored treatment? Can we help people? How many are out there? Because right now, people aren't being screened for infections, and I think that's a problem. So what else are we doing? We're sharing samples. A sample of someone's brain, that's a life. We should be utilizing it to its fullest extent. We shouldn't have it at one lab, so that it can have narrow analysis with metabolomics and then write a paper on it. We should do multiple different methods, looking at the host response, characterizing those tissues, looking at microbes, looking at pathology, look at all of it on those same samples. We're going to do that. We're publishing high impact reviews to harness the wealth of data that's already in the literature. So our consensus paper was the first one, and our second one is in press and will be out soon in a special issue of the Journal of Infectious Diseases, all about how microbes invade the sensory system and get in and also lead to sensory deficits and damage. We also are inspiring younger scientists and welcoming them into our midst. This is not just a whole group of people that's the same. We have junior people, we have senior people, and we want to create opportunities for younger folks to be both mentored and learn and grow within AlzPI. And then finally, education and awareness, things like we're doing today. None of this at all is possible, and I'm actually impossible for me to be here today without some of the people in these pictures and some of the people sitting in this room. Because literally, the research that's done on microbes and infection, even if it doesn't make its way into a guideline somewhere, and change practice behavior on a broad scale yet, doctors like Chuck Stratton, who treated me, he was harnessing the literature. He was saying, Dr. David Hahn, your work on asthma, that's going to inform me putting Nikki Schultek on azithromycin for her asthma, right? So the work means something. It means something to people like me. And this team has been incredible. And unfortunately, we don't see each other that often in the flesh. It's mostly on Zoom. Some of us for many years, like Lavinia and I in the back row, worked together for 5 years before I saw her in person because she's in Switzerland. But this whole thing is possible because of our incredible team. And there are kind of a lot of us, and people are working with us in various capacities. Some people are sharing samples, data. Some are actually going to be study sites. Some people are going to just publish reviews with us, and others are going to help in other capacities, and we're always open to more collaborators. And finally, and definitely not, last but not least, I guess is the way to put it, we are so grateful for the funding support from these three organizations, and especially to the Sim Einstein Research Foundation Incorporated. Steve and Marilyn, your support has been incredibly unbelievable and generous, and it is giving us a chance to generate data to help us help people. And the same thing goes for the Godsnell Foundation and to All's Out. Thank you, Chad. I know you're back there today. Thank you, Kate, on Zoom. We're so appreciative. So with that, I conclude. I don't know, I'm early, right on time. Okay. I can take, what do we have, like 1 minute? If anyone has a question, I think we have like an extra minute. Oh, 5 minutes? Okay. Questions? Anyone? I think in some cases, for me, absolutely. In many cases, yes, but in some cases, if it's unpublished data, possibly no, but that's a great question. Yeah, absolutely. Marilyn? Yes, hi Sandra, I hope you're on. I did send Sandra the Zoom link this morning. Yeah, absolutely. And thanks to my family who's watching at home. And thanks to my brother for coming and Dave for coming in person. If nobody has anything else, we can just continue on with the next presentation. Thank you.
Thank you, Nikki. That was wonderful.
Thank you, Brian.
find where I am. Okay.
I'm gonna,
Okay, I have.
just a hot. I'm still on. Okay. Can everybody still hear me? Okay, good.
Yeah.
Sorry, I'm having like tech issues up here. Or I'm not having tech issues. I'm having a lack of confidence in my tech, which is clearly working. So I just want to say something really quick about Brian. So I met Brian at the very beginning of my journey. So I'm just going to give him a brief introduction that he really deserves. Brian's one of the most wonderful people I know. He's my friend. He's my colleague. We've been working together for now 7 years. He works to help people. He is a true collaborator. He is our fearless leader on our grant writing efforts. On this symposium, he has been just the most wonderful work partner to join forces with. And so with that, I turn it over to Dr. Brian Balin.
Thank you, Nikki. Much appreciated too. I'm just gonna take this. I just wanted to make a quick mention too that all of the individuals that are participating today virtually, if you have questions, please put them into your chat and we'll try to answer those questions along the way, especially when we have a break. So my presentation today is evaluating infection and Alzheimer's disease pathogenesis. And I first wanted to start out with acknowledgement. And you see on this slide, I have quite a lot of students that have participated with this effort. And I have, from my own laboratory, Dina Pelt, Chris Hammond, Scott Little, Sue Hingley. There's a whole list of individuals that have participated and continue to participate in the research that we've been doing here. The other thing I wanted to make mention to is of a few different people that are listed here and some in the audience. Alan Hudson and Judy Whittam Hudson. Alan and I first stumbled into asking the question about infection and Alzheimer's disease in the mid-1990s. And we were just having a conversation about an organism, Chlamydia pneumoniae, showing up in atherosclerosis. And we simply asked the question, could that also be involved with brain pathology and with Alzheimer's disease? And the reason we went down that path is that my background was with studying the blood-brain barrier and how things actually get into the brain from the blood. And knowing that atherosclerosis, even at that time, this was mid-90s now, was being considered as a risk factor for Alzheimer's disease, we just asked the question. And it led to really an evaluation of brain tissues that had been collected over time and finding evidence in the brain tissues for this organism. So I thank Alan and Judy and Hervé Girard and their group for that early work as well and really driving this along with me to hopefully make a difference. The other is Todd Abrams that's listed here. I went back to my laboratory after talking with Alan and talked with Todd Abrams, who's an immunologist, and said, okay, how could we possibly detect this organism in brain tissue? And what's happening immunologically? And so we started talking about it, and then we came up with a way of actually taking homogeneous of brain tissues, putting them in culture with cells in culture to see if we could grow any organisms out in those cells. And the reason we did that was that Chlamydia pneumoniae is one of those weird organisms that's an intracellular bacterium. It actually invades our cells and grows there. It has to. And I'll talk a little bit more about that shortly. But it led to literally growing out of a few different Alzheimer brain tissues, this organism, and showing it by both immunolabeling and electron microscopy, et cetera. The other person I wanted to make mention of here that isn't on here is in the audience, is Ken Blank. And Ken has been a supportive of ours from day one, and we were working at the same institution. And when I talked to Ken about this, he was immediately supportive and understood what we're talking about. He also is an immunologist and really understanding what it is we talk about here when we have infection on board with an immune response as well. The other I wanted to make mention here, supported by. Okay, There's a couple of lists here. There's an NIH support. There's an Alzheimer's Association support. Very limited support over the years. But I wanted to specifically mention these foundations. The Adolph and Rose Levis Foundation, Foundation for Research into Diseases of Aging, the Essel Family Endowment Fund, the Osteopathic Heritage Foundation, the Sim Einstein Foundation, we're here, and the Pia Stratton Fund. When Charles Stratton's wife died, he set up a fund to come here in the name of his wife. And now, when he is now deceased, this is a fund we're using to study this effort. So I appreciate all of these funds in particular, because that's how we've continued our work. It has not been through our national agencies or the overall Alzheimer's Association to any great extent. It's through these funding mechanisms. So I appreciate that and thank all of these organizations. Now, learning objectives. What's the infection hypothesis of Alzheimer's disease? What are the approaches that we can use to study the host-pathogen relationships? And a perspective on what we've been looking at with Chlamydia pneumoniae. And that's going to be more in a summary fashion, but it's going to give you a picture of why we think it's an organism that we need to consider in this analysis. The major risk factors for Alzheimer's: aging. Number one risk factor. As we get older, we run the risk of now developing dementias and Alzheimer's dementia and other types of diseases as well of the nervous system and other chronic diseases, obviously. But genetics is also a factor. We can't get away from that. We have to consider it. And in consideration, one of the big genetic risk factors is an apolipoprotein. So we've heard about these apolipoproteins, E and the epsilon 4 factor for the most part. But there is a genetic form of Alzheimer's disease as well. That is a familial form, hereditary form, that actually is due to genetic mutations. In late onset sporadic disease, we don't see those same mutations. But in very early presentations, if you evaluate the families, and it would be like a pre-senile dementia type of presentation, you do see mutations in the beta amyloid precursor protein gene, in the presenilin genes 1 and 2, et cetera. And there are other genes too that have come out of the woodwork that may be also participating. But those three that I mentioned really are involved with familial disease. The apolipoprotein also gives us risk factor though, and it's normally made. We have different forms of it in many of us, all of us in essence. And you can get different risks from that. But I also list the other risk factors over here to the right. Atherosclerosis, accepted risk factor. Diabetes, accepted risk factor. Head injury, brain trauma, now has come onto the scene and also a big risk factor. Toxins, possibly. We're still, in a holding pattern with toxins, but there is one that comes from cyanobacterium. The beta methylaminoalanine, that's cyanobacteria. Cyanobacteria, by the way, are blue-green algae. They're in all of our water supplies. We can't really get rid of it, so we're exposed to that. So is that a possibility as well? We have to consider it. And then, of course, infection that we believe is a big consideration. I just have this a slide up in summary so that you can see that Alzheimer's disease and dementia, the signs of that is different from the typical age-related changes. We're always getting questions of, oh, geez, I'm having a word search issue in the older population. Is that Alzheimer's disease? Or I've forgotten somebody's name or misplaced something, just as a matter of fact during the day. That's not Alzheimer's disease. That's not dementia. That's sort of part of the normal aging process. But Alzheimer's really is now a big change where you're now getting a chronic change that is consistently chronic and occurring. Alois Alzheimer studied pre-senile dementia, identified plaques and tangles, also studied neurosyphilis. Neurosyphilis is something that will cause dementia. Okay, so that's well accepted. There are other organisms that cause dementia that get into the brain, such as HIV. In late age, HIV entering the brain can also lead to a dementia. There's sometimes you can find measles virus, the odd measles virus that is mutated, shows up in subacute sclerosing panencephalitis, also can lead into a dementing illness. So we already know that these things can happen with infectious agents, and now we're just expanding upon it. I wanted to make a point here about the pre-senile dementia. The individuals that Alzheimer's was evaluating, these were younger individuals for the most part, in their 50s for the most part, some in the 40s, but in their 50s. It was a pre-senile process where he found these plaques and tangles. The other person I want to make mention of, because it is completely relevant to our story today, is Oskar Fischer. Oskar Fischer, this Czech academic, psychiatrist, neuropathologist, he was actually studying senile dementia, showing that there were plaques and tangles, and he actually is the one who identified senile plaques in brain tissues. And there's a difference here. Senile dementia, pre-senile dementia, okay? The pathology is almost identical, but you've got two different diseases in essence. And what has happened since about the late 1970s, early 1980s, is an acceptance in the research world of Alzheimer's disease that because you have plaques and tangles showing up in hereditary Alzheimer's disease, mostly pre-senile dementia, and also in senile dementia, that it's the same disease. This is not the case. There are many forms of Alzheimer's disease under this umbrella, and we have to regard it that way, and we have to start thinking about, well, what's the process of infection in this, especially senile dementia, because that's where we're starting to see more of the infections showing up. By the way, he was also studying neurosyphilis, and at the time, big issues about tuberculosis, mycobacterium organisms. This was being bantered about. This goes back to 1907, 1912, the early 1920s. We are over 100 years later here, and now still trying to figure this out, but really on the path now to doing it. So what's initiating these senile plaques and sporadic Alzheimer's disease? Obviously, the amyloid cascade hypothesis has overwhelmed the field. Now, there's also a tau hypothesis. You can go to the ApoE issues as well. There are a lot of different issues here that you could go to, but the amyloid cascade hypothesis overwhelmed the field, suggesting that amyloid is bad stuff. It accumulates in plaques. It leads to tangle formation. It leads to Alzheimer's disease. The other part of this is neuroinflammation. And neuroinflammation came about, the big studies with Sue Griffin and Joe Rogers, Pat McGeer in the 1990s. It took them a decade, a full decade, to be appreciated and accepted in the Alzheimer world that neuroinflammation was key to the disease. So now we have this basically merge happening here. Yes, amyloid is important in the disease process. Tau is important. ApoE is important. Other aspects are happening here. However, microbes, when you throw microbes into the equation, you go, okay, it can lead to amyloid. It can lead to neuroinflammation. You can then have now this triumvirate occurring at the same time. And also, you can have tau forming, et cetera, and neurodegeneration now progressing. The infection hypothesis, microbes trigger the inflammatory response. You get activation of neurodegenerative responses and pathways, beta amyloid processing, beta amyloid deposition, tau neurofibrillary tangle development, and then we call it Alzheimer's disease. So that's just a very basic designation of what we're talking about here. Proposed infectious agents. So here I list bacteria, a number of them. Today we have with us individuals that are going to talk about these different types of bacteria as well. The Porphyromonas gingivalis issue is very big and very huge here. An anaerobe organism that causes periodontitis. I'm going to talk a little bit more about the chlamydia organism, viruses. Obviously, Ruth-Saki came out in the early 1990s with the herpes simplex virus information, and then basically struggled to get people to accept what she was talking about. Even before that, Alan MacDonald started talking about Borrelia and Lyme disease. And then Judith McLaughlin started talking about Borrelia burgdorferi and Lyme disease again. And we stumbled into the chlamydia work through total serendipity in the mid to late 1990s. Fungi. So we're going to hear from David Corry today and his group from Baylor, Lynn Bimler as well, about Candida species involved. We're not really going to hear today about Toxoplasma gondii, but here you have a parasite that also can be involved with Rima McLeod's work. And then you have other oral, gut, and other organisms, because this is a wide open area, an arena here, because we're still finding different types of pathogens showing up in brain tissues and in fluids from individuals with dementia. So investigating the mechanisms and outcomes of host pathogen interplay, in vitro approaches and in vivo approaches. This is what we're talking about here. All of these types of approaches now are being utilized to try to now understand and using now methodology, proteomics, metabolomics, transcriptomics, looking at pathology, looking at molecular mechanisms here. All of this is important. And many of us have shown over the years, mechanistically, how some of these organisms are contributing to changes consistent with what we find in Alzheimer's disease. So the Chlamydia pneumoniae story, it's typically a respiratory pathogen. It causes community-acquired pneumonias. It's been involved with asthma as well, chronic obstructive pulmonary disease. So a lot of these diseases, including atherosclerosis. If you look in PubMed, you will find there's over 1,100 articles published in PubMed on its relationship with atherosclerosis. And many of us believe that it is one of those factors that could actually cause atherosclerosis. So this is an intracellular bacterium. It requires ATP for growth. It requires nutrients for growth, et cetera, from our cells. So it's growing in needing this. There's serological evidence over the years, up to about 75, maybe even 80% of the population as we get older, we've been exposed. So we have exposure. Do we have disease from it? Not necessarily. Some people can get significant disease, we think. Others may harbor, and others may have just been exposed and reacted to it, and it's not doing anything. So we have this wide variety of outcomes, possibly. This is what we came out with the first paper that we published, this identification and localization of the organism in Alzheimer's brain tissues. Over here is a neuron. All that brown in there is a big inclusion labeled for Chlamydia pneumoniae. And at times you'll find that. Other times, if you do the electron microscopy, which over here is to the left, from Alzheimer's brain tissues, these are what we find. These are the dense bodies here are elementary bodies, and the light ones here are reticulate bodies. And we found it initially by PCRing, and then we started looking at immunolabeling in brain sections and found that, oh, you have infection, you have pathology. That was pretty cool. And then, of course, I mentioned earlier that we've even cultured it out of a number of different Alzheimer's brains. And the culturing methodology is this. B and C show you now the organism present within monocytes in culture, and D and E are the electron micrographs of those cells. So we put it under and look at the ultrastructure, and you can see that these organisms show up inside of the monocytes, which is really interesting. That led to a lot of other studies that we list here. But I wanted to make a mention about monocytes for a moment and finding that this organism in living beings can also now be engulfed from the lungs by our white blood cells, monocytes in particular, could be neutrophils as well, and also there's evidence for lymphocytes that can pick the organism up. Well, that suggests then the organism is in blood, can be in blood inside of cells, can circulate around. We think that's why it's involved with atherosclerosis. But what's interesting here is if we go back into the literature, in the late 1980s, the Manuelides group was looking at an infectious component in blood that they thought was causing Alzheimer's disease. And what they did was they filtered blood. They didn't use white blood cells. They filtered the blood and took the filtrate. It did the filtration through a 0.22 micron filter, took the filtrate, injected it into animals to see if you could now actually infect an animal with that filtrate. It didn't work, but they were on the right track. What they didn't think about is, oh, what about the white blood cells? Maybe there's something in white blood cells that could be really important in this disease. So up here, what we've studied is routes of entry, nasal olfactory epithelium, the olfactory system, really a big issue here with how we get infected. The blood-brain barrier as well, obviously anything in the bloodstream. Brain areas that we found: frontal cortex, entorhinal cortex, hippocampus. So the entorhinal cortex, hippocampus, amygdala, all of those are limbic structures in our brains. They're all connected to olfaction. What we've proposed for many years is that if you get infection through your olfactory system or insult to your olfactory system, it can lead to insult into the limbic regions of the brain: amygdala, entorhinal cortex, hippocampus, even crossing over to the orbital frontal cortex through the anterior commissure. Okay, where does Alzheimer's disease start? It starts in our limbic system. The entorhinal cortex is really the key area where this starts. So is olfaction important? We absolutely think so, as a big issue. And we're not the only ones. Richard Jody also thought this, published on this in the late 1980s, throughout the 1990s. Another collaborator of ours, Dev Devanand, also, and I have a slide coming up attributing a quote to him, that also found that olfaction and our ability of smelling actually could be affected here in this disease. And it's affected also in other diseases as well. Other cell types, by the way, that we found, glia, neurons, and endothelial cells, could be infected. So noting that we can find something in white blood cells, we actually did a small study looking at samples from AD and MCI patients, took their blood, spun out the white blood cells, the buffy coat, evaluated what's in those white blood cells, and found evidence for your elementary bodies and reticulate bodies for the most part, and the blue bars there a little bit with persistent bodies. This organism, when it infects cells, can also lead to an inhibition of apoptosis, meaning the cells are long-lived that contain the organism, which is a big problem, because now you have potential persistence. So this is how we actually think that a brain like this, through Homer Simpson, could actually be infected, but also through the lungs and the bloodstream. So this is a big key to us in studying these interactions. And I think we need to study both, because in some cases, it could be blood-brain barrier first in the hippocampal region, for instance, or could be olfaction first, or it may be happening together simultaneously, where we're getting those changes leading to the early changes in Alzheimer's disease. This is the quote I wanted to mention from Dev Devanand from 2000 in the American Journal of Psychiatry. The findings suggest that the inability to recognize smells combined with a lack of awareness of impaired odor perception may be a sign of impending Alzheimer's disease. We have clinicians in the room. We have clinicians online, I'm sure. I've talked to clinicians. How many do smell testing for their patients early on if they come in and they have some kind of cognitive change. How many do that? And why shouldn't we be doing that as using part of our diagnostic? The other thing I wanted to just quickly show you is that chlamydia pneumonia infection promotes increased intracellular beta amyloid 1 to 42 in astrocytes. This is work that was led by Dr. Appelt in the laboratory and my great colleague for years and years. And what they're seeing here is that with chlamydia pneumonia infection in green, you also have increased in red. And the red is labeled for the amyloid that's showing up in astrocytes now in higher levels. And if you evaluate, which they did, over a 72-hour period of time, what's happening with the enzymes that are involved with creating amyloid peptides, you see that beta secretase and both the presenilin 1 actually are elevated here in green compared to uninfected cells, which is really interesting, suggesting you have a mechanism affecting the particular proteins that are actually cleaving your beta amyloid precursor protein into these abnormal forms. The other thing I wanted to make mention of here is the relationship between other risk factors and this infection. The ApoE story, which the Hudson group really was working on significantly, finding that with ApoE epsilon 4, it will actually enhance attachment of Chlamydia pneumoniae to cells. So in the presence of ApoE4, you can actually have an increased attachment, which is really interesting because it would lead then to increased infectivity. Atherosclerosis, all of these different studies which I mentioned. Diabetes. McLossy and McGeer actually did a study in showing that pathogen burden showed a strong association with insulin resistance. People have been questioning, well, why do we have insulin resistance in diabetes? And why is diabetes related to Alzheimer's disease, which some people think is a type 3 diabetes? Well, we think it's because if you have infection on board, you get insulin resistance. And that's where you're not utilizing glucose the way it should be in the nervous system. And then of course, with neurotrauma, we have a whole variety of issues here that not only where chlamydia pneumonia could be involved, many of the organisms we're talking about today could be. So this is now a model of first starting with infection and invasion, with invading the neuroepithelial cells, olfactory neuroepithelial cells, the blood-brain barrier, distribution in monocytes, going then into cellular dysfunction. What I haven't shown you today is all the different dysfunctions that this organism can lead to, anywhere from mitochondrial changes and generation of reactive oxygen species to calcium dysregulation, iron dysregulation, interferon gamma production in the brain, et cetera. There are many pathways that we've actually investigated. And then the final thing here would be neuroinflammation, where amyloid, we think, is getting deposited and made first, even before it's deposited. It's having an effect. It could be having an anti-infective effect as well, an antibacterial effect or antiviral effect. But also, as it starts accumulating, it's going to accumulate into these plaques. And we know that microglial cells get activated, astroglia try to wall off, and leading to more and more inflammation. But what's really the trigger and inciting agent for this? We think the chlamydia pneumonia in this case and other organisms. So thank you. I don't know if we have any time for questions, but we have 1 minute. Thank you. Yes, David. Thank you. Yes, oh yes. So how prevalent is chlamydia pneumonia in Alzheimer's disease versus non-Alzheimer's disease? What we're finding is in our analysis, we found up to about 10% of brains that we analyzed. Okay, we've analyzed a number of different studies, probably close to 100 brains, I would say. That up to about 10% in non-Alzheimer brain tissues, we could have that detection. 90% of the Alzheimer brains we looked at, 90% actually showed that we had infection there. Now, showing infection there obviously isn't necessarily causation. However, we believe that based on our analysis that we've done a lot in culture work, et cetera, and animal modeling too, which I didn't really even talk about today, you can generate the pathology, or at least part of the pathology, and the disruption of the pathways. And I think that those individuals where that's happening, they're now going on to have cognitive change. And then, so it could be harbored and not and persistent and not really doing anything, and in others, though, something drastic. Yes.
Brian, given the probably get they actually jumping from that to the nerve cells in the brain?
So what happens with this organism is that there is a level of release of the organism from infected cells, even though it will inhibit apoptosis. It has been demonstrated that this organism can, at times, kill a cell. It can be released, so you can get a humoral response to the organism. That's how we're getting that, basically, through processing to some extent. So it is a possibility that once it's secreted in, it can now be picked up. Now, if going through olfaction, you could already be in your olfactory neuroepithelial cells going into neurons in the olfactory bulb, picked up that way.
Brian, do you
So-
mind repeating Dr. Shee's question for the online people?
Oh, sorry.
No, no worries. I just, we got feedback. on it.
Okay. For the online people, the question was about whether Chlamydia pneumoniae coming through monocytes, for instance, how would that then lead to infection of neurons in brain tissues? And that's how I answered that question, is that it can be released from those cells as well and picked up by other cells. Yes.
So the question is, Ravi, if I'm understanding, are all strains of chlamydia pathogenic or are some of them not?
Well, it's a good question. So it's a good question. I would say at times the organism appears that it can become persistent inside of a cell. With persistence, is it pathogenic? Is a good question. If it's actively reproducing, it seems that the cells are being activated in many different ways. You're getting inflammasome activation, pro-inflammatory processing, and it seems that they are then quite pathogenic. But with persistence, we're not sure. And persistence may be a big issue here so that it can hang around for a long period of time and then activate due to other factors as well. Okay, so I think in the order of keeping with our timing, I will now introduce Garth Ehrlich and Michiko Thwe, who from Drexel University, who will present their information too.
While they're getting mic'd up, I just want to let everyone online and here know that we'll have some time for Q&A as well after each session.
So I'm Garth Ehrlich. I'm going to tag team with Michiko, who's my graduate student. And so I'm going to start with presenting the assay that we've developed and have been using to look at Alzheimer's brains. And then Michiko is going to talk about actually the results that we've been getting, with this assay. I want to start by noting, as Brian did, that trying to get this work funded through traditional channels is very challenging. We've been very fortunate. We've had very substantial funding through what's called the Oscar Fisher Project, which is a philanthropy that James Truchard started, and also from the Bill and Marion Cook Foundation. Nina Tentet is here today, actually from the Bill and Marion Cook Foundation. So I want to really start out by saying thanks to our funders. So one of the hats that I've been wearing for most of my 40 years in the field of infectious disease is molecular diagnostics. And so to me, having the best assay we could have is probably the most important thing. And so we have been, over the past 8 years or so, been developing what we call pan-domain assays. And so a domain would be like bacteria or fungi or parasites. And we now have a trans-pan-domain assay, which will provide species specificity essentially for any cellular pathogen. And so I'm going to talk about how we got there, kind of like. So we use long PCR, and then we do long read circular consensus sequencing, and I'll go through how this works. The only declaration I have is we're actually developing these assays for Drexel Medicine Diagnostics. So as I said, the concept for a pan-domain diagnostic is to provide unparalleled sensitivity, specificity, and breadth within an entire domain. And so that you have an unbiased way to survey for everything within that domain that could possibly be in any specimen you're looking at, regardless of tissue type or specimen type. And so just to kind of summarize, what this does is it allows you to look very deeply. You don't need to use culture, so you're not reliant on the organism's ability to grow. You can identify mixed assemblages of microbes, polymicrobial, or, consortia. It's at least semi-quantitative so that you can get the relative levels of all of the organisms that are in your specimen type. And of course, because it's species specific, we have very high resolution genotyping. In some cases, we can even do strain analysis if there's different ribotypes. And you can also identify, if you will, emerging agents that have not been previously seen. And I can tell you with our eukaryotic pan-domain assays that in some cases, the majority of organisms that we find no one's ever seen before because the databases just don't exist. So once you've got a pan-domain assay, it becomes very cost effective because even if new agents are discovered, you're covered, okay? And we can multiplex it so we can do it in a high throughput manner. So just to kind of review, it doesn't require cultural organisms. It can work following antibiotic therapy. If you're looking at a patient, you can detect biofilm bacteria and fungi, which are again, could be often difficult to culture. You can detect multiple species simultaneously, provides relative quantitative data, and as I said, it reduces workload because there's no need to develop a new assay for some new organism, that has emerged as a potential associated with any sort of disease you're looking at. And at least for the bacterial pan-domain assay that we've been using, it's highly validated. We've had numerous studies using it in multiple different fields, not just in Alzheimer's disease. And the work has been published for a number of years with hundreds of citations. So the original microbiome studies were actually done based on the 16S ribosomal gene of prokaryotes, bacteria. And they really revolutionized community-based studies because they revealed unprecedented levels, of complexity in natural systems and organismal systems, what we call the holobiont, which is a metazoan in their microbiota, plants in their microbiota. But they suffered from a lack of specificity because when you were doing the original next-gen sequencing, you're sequencing very short regions. And so you oftentimes can't get below family level. Sometimes you get genus level, but you oftentimes can't get to the species level. And we've been working with Pacific Biosciences, PacBio, which is a third generation sequencing technology, which provides for very long read sequencing. And so we've been beta testers with them pretty much since they started in 2011. And I'm not going to go through, what's behind it, except to mention that it's really revolutionary. There's new physics involved, there's new biology involved, and there's new chemistry involved compared to even, the standard next-gen type sequencing, which many people are familiar with, such as Illumina. And in the later generations, it's also extremely high fidelity. And this is where we get our species specificity from. It's called single molecule real time. And it provides for single molecule error correction, which almost sounds like an oxymoron. But it works through circular consensus sequencing. And this is really, really clever technology, so that each sequence gets read multiple times. So you have some error, random error rate. It doesn't make the same error on each pass. And I'll just kind of go through this a little bit in the next slide here or two. So each base is read by the polymerase multiple times, provides for extremely low error rates. And so we're at the point now, actually, when you're doing circular consensus sequencing, that it's as accurate as Sanger sequencing or more so. And so this is the idea. If this is our full-length 16S gene here, the gold and purple PCR, you add these, what they call these smart bell primers, to a play on dumbbells, at the ends of the ampler. And then you add in a primer, and now you've got a partially single-stranded, partially double-stranded region here, which of course is recognized by a DNA polymerase. And so the DNA polymerase will start and go around, and it'll just keep going around and around. So for each pass, you get these subreads. You add and then you can align all the subreads, and any mutation that's only in one read just gets averaged out. So we ended up with like Fred scores of like 43 and 44 now. And this really has kind of, between the long reads and the circular consensus sequencing, has really revolutionized molecular diagnostics. And so we have two pan-domain assays. The first one is the U-bacterial pan-domain assay, where we sequence the entire 16S gene, which is about 1.5 kilobases long. And more recently, we've developed a eukaryotic trans-pan-domain assay, where we're actually amplifying about a 3.5 kilobase region of the eukaryotic ribosomal operon, which includes the 18S gene, ITS1, 5.8S gene, the ITS2, and then into the 5' end of the 28S gene. And then, if you've done these long amplifications, then you put a barcode on for each specimen, and then you put the smart bells outside of that. And so this allows for multiplexing 100 or more specimens in a single smart cell on the PacBio instrument. And so this is just the complex algorithm we have for analysis. This is all published. I'm happy to talk with anybody who's interested in this later on, but I'm going to kind of skip over this. And one of the other things that we did is we realized that although most people have used average error in determining how to analyze sequence, Josh Earl and my group actually showed that using expected error is a much better analysis method. And again, I'm not going to go through the arithmetic on this, but I'll show you the results. And so using a BEI generated mock microbiome, where we knew there were 19 bacterial species in it, we showed that if we use only those reads that have one or less expected error to form our initial bins, which are essentially the species, they're called OTUs, we would get exactly 19 species. If we allow even 2 expected errors, we start getting more OTUs. And this has, of course, been the problem with microbiome studies since they started, is they tend to overrepresent the number of bacterial species or whatever species you're looking at that are actually there. And so this is actually the bacteria that were in that BEI challenge set, and with one exception, that each one's from a different phylum. So it provides for very, very broad coverage of the bacterial domain. And we were able to identify each of these with 99% confidence in every case. There were 2 organisms that were in there that we missed, and we knew we would miss them because one was Candida, which is a fungi, and one was an archaea. And then there was, I said, there was one set of very closely related organisms there. They had both Staph aureus and Staph epi. And from a genomicist's point of view, they're actually a single species because they have over 99% 16S sequence similarity. And generally, we define a species genomically with SEO of only 97%. But because these are so important, we actually added a wrinkle in our algorithm to be able to distinguish them based on SNPs. Then this is kind of my last slide or two. We then got a challenge set from the Joint Genome Institute out in Walnut Creek, which was not expected to be able to be handled using ribosomal analysis. It was designed for people who were doing massively deep metagenomic sequencing on like an Illumina 10X, and there were over 250 bacterial species in this challenge set. And we did better than anybody using a 10X. We were able to correctly identify almost every organism that was in this group, and we actually corrected a number of taxonomic mistakes that the organizers had made when they were assembling it. And I'll just show you one of them. This just shows the linearity of our assay over multiple orders of magnitude. So the diagonal line that would actually be perfect, and all the dots are how we found the organism, relative to, what the ideal was. Of course, we get some scatter down at the lower levels, but overall, it's pretty linear. And so within this set, there were supposedly 14 different Clostridial species, and we only found 13, so we were kind of disappointed. We missed this Clostridium bartlettii, but actually we didn't, because Clostridium bartlettii had been reclassified by Burgess as now Intestinobacter bartlettii, and we found Intestinobacter bartlettii. So even amongst 250, other species, we were able to get species identity for all 14 Clostridial species that were in that challenge set. And so you kind of want to leave you with this, this is why we have great confidence, in this assay to be able to essentially determine any organism, that is in any specimen type. I'm going to turn this over now to Michiko, who's going to talk about the work that she's been doing with this assay on our Alzheimer's brain specimens.
Thank you, Dr. Garth Ehrlich. Good morning, everyone. My name is Michiko Thwe. I'll be talking about uncovering the etiological roles of microbes in Alzheimer's disease. So recent studies have shown that amyloid beta peptide is an antimicrobial peptide. That has been talked by Dr. Brian Balin talk as well. And we believe that amyloid beta peptide are the one that try to entrap and neutralizes this invading pathogens in the brain parenchyma. So with a repeated stimulation via chronic infection, it could result in overprotection of amyloid beta peptide, forming into a plaques, which became one of the hallmarks of Alzheimer's disease. So with the growing body of evidence, we started to suggest this new AD pathogenesis called infectious hypothesis, where we believe that when there's a microbial infection, that might be a bacteria, virus, or fungi infected in the brain, making the amyloid beta plaques to overproduce, resulting the tau protein to hyperphosphorylate, and because of that, it leads to local neural inflammation, causing a neuronal cell death. So we believe that microbes may play either or both etiological and X-ray role in the development of Alzheimer's disease. Oh. Better? Oh. Can you hear me now? Good. Okay, great. So from our preliminary data that was recently published by our lab from Frontiers, we have detected in the bacteria in the brain, including Bacterium acnes, which was previously known to be a Propionic bacteria acnes, P. acne, Methylobacteria, and Acinetobacter, and Comamonas using our full-length 16S RNA gene-based assay. There has been studies out there that talks about fungi such as Candida and Botrytis species observed in AD disease brain, and viruses like HSV1 and zoster has been suspected to play a role in Alzheimer's disease. So when we talk about brain requests and looking at the specific brain region, there are several regions that we're very interested in looking into. Starting off with frontal lobe, because it has a huge memory processing area. Temporal lobe due to its station of hippocampus. Olfactory bulb is another region that has no blood-brain barrier or very lower permeability of the blood-brain barrier, and it's very closely connected to entorhinal cortex. It's a similar slide that Dr. Brian has already presented, but so we are looking for the specific brain regions to understand more about the microbiomes. Lastly, we wanted to look for primary motor cortex and hippocampus as it shrinks a lot in Alzheimer's disease brain. The conditions that we're looking into are Alzheimer's disease versus healthy control. Currently in our lab, we have age match control to do a standardized protocol, and we're going to be doing 16S or 18S sequencing at the end. That means that we are using a frozen brain tissue instead of the brain fixed one. The ideal brain tissues are usually post on an interval group of 0 to 5 hours. And currently, these yellow highlighted ones are the ones that we have in our lab. So this is a typical Alzheimer workflow of day one. Starting off with the DNA extraction, the only different part of brain tissue compared to other regular human body tissue is it's very sticky on surface. So when you use a brain tissue inside let me check here. It shows a bigger bucket here. It's actually a bigger Petri dish where you have to add in dry ice to control the temperature throughout the processing so the brains are not melting in the process of sample preparation. And I'll explain a little bit more about edge versus core in the future slide. So after that, we will proceed with DNA lysis and incubate it overnight for the day 2 of extraction. Here at this part of lower bottom, we are just starting to develop a FISH technique, which is called fluorescent in situ hybridization to visualize the microbes in the brain. So this is one of the example that we have for the brain tissue, where we'll try to trim down the brain to get the edge core analysis to rule out any contamination. So this is another study that we have done to make sure that after trimming of this particular part, we could be able to do a biopsy punch in the middle to get the centerpiece of the brain tissue. And this is what I was talking about before, where you have to use a dry ice to make sure the temperature was controlled throughout the process. Alzheimer's workflow day 2 is now similar to other human body tissue extraction. You just do meat beating and several washes to get the purified extracted DNA. So 16S sequencing is the 3rd day or after the 2 PCR gene amplification step, where you try to do 16S RNA gene contains of the 9 hypervariable regions with the conserved region. That means that these 9 hypervariable regions are the one that's distinct towards bacterial species, and conserved region will have similar across all the bacterial species. We'll be doing a PacBio sequencing that will generate longer reads, which able to sequence nearly entire 16S RNA gene covering all these 9 hypervariable regions. With that, we could be able to get the raw data using a CCS via PacBio, where we can then be clustered into operational taxonomy units based on the similarity of sequence, which is then mapped to using an MCSMART pipeline that was developed from our lab. So this is a result that we have received from University of Arkansas Medical System Brain Bank. This is the top 30 of overall genera species that was found overall prevalence among all specimens, starting from top to bottom, left to right. And I just wanted to point out the first top 2, Acinetobacter junii and C. acne. These 2 had the highest prevalence that was found out of 30 brain subjects that has 16 of Alzheimer's disease brains and 15 of control brain. And it was done edge versus core protocol. So 31 samples of them are edge and 55 samples are core. And it was done in 4 different extraction days. So with the 4 extraction control found it. And most of these bacteria are commonly found in soil and sewage water, which is very surprising to see. So one of the data analysis that we have done for microbiome is Dirichlet multinomial model, where we'll try to quantify the relative abundance between 2 sample groups. By looking at that, we look into Alzheimer's disease versus control group, and we also look at edge versus core. The reason we look at edge versus core is to rule out the contamination, and we wanted to verify our published data that was published back in Frontiers. And we found that there's no significant difference with edge versus core. That means that the relative abundance that was found in the edge surface of the brain versus the core, the center of the brain, is the same. So when you look into relative abundance across all the samples comparing AD versus control, we have found 10 species that's relatively different, and 6 OTUs or species that was mostly found are C. acnes, Acinetobacter J, and so on. For control group, I wanted to point out Acinetobacter junior that was show up again in the control brain, along with Novosporoquium and Morozella. When we look into only the edge sample, we have found 6 OTUs species again that was showed up, and only 1 species that was show up for control, and this is Acinetobacter junior. Lastly, when we're looking only for the core sample, which is like a totally center of the brain tissue, we have found C. acnes again with Acinetobacter johnsoni, and Morozella is a new one that was showed up for the control group. To summarize from our data, we have detected more C. acnes associated with Alzheimer's disease and Acinetobacter junior associated more with control group. And I would like to thank Bill and Marian Cook Foundation and Oscar Fisher Project to help fund this project and able to do more future projects as well. And I would like to thanks to Drexel University, especially AlzPI, to able to speak in front of here, and Genomic Core, Dr. Yves Monet, who is in the audience right now to help analyze the data, and all these neurobiobanks, University of Arkansas, University of Washington, Wisconsin, and thank you.
Thank you, Michiko. That's wonderful. And Garth. Our next presenters are going to be Dr. David Corry and Lynn Bimler from the Baylor College of Houston.
We good? Yeah, looks like we're live. So good morning. Thank you very much, Brian, Nikki, and the other organizers for a really great meeting, for an invitation to speak. If I seem a little slow, maybe I misspeak a little bit, you can thank Garth for that and all the great wine that we was pouring into my glass last night. Thanks, Garth. It was really good wine. Okay, so let's get going here. Lynn and I want to talk to you about our work now with the fungus you've already heard about, and that's Candida albicans. Let's see if this already have an electronic. Maybe I'm hitting the wrong button. I could stand back here. Sure. There we go. All right, so we've already heard about many of the microorganisms have already been implicated in Alzheimer's disease. So beginning with herpes simplex virus, and there's chlamydia pneumonia, we've already heard a great deal about. And then there is the various spirochetes, including the agent of syphilis, Treponema pallidum, there's Borrelia as well, that have been implicated. There is emerging data with the gastric pathogen Helicobacter pylori. And then, of course, and quite a few other organisms. But I want to spend our time today talking to you about an organism you might not have heard that much about, and that is the fungus you've already heard about, Candida albicans. So beginning in 2019, we published our original work with this model of Alzheimer's-like disease in the mouse, in which we inject 25,000 yeast phase cells of Candida albicans intravenously into mice. And what we get are images, for example, such as this. This is a whole mouse brain. And you can see with our staining for IBO1 for activated microglia and GFAP for astrocytes, you can see these about 50 to maybe 200 micron spherical structures There's only one in this view in the entire mouse brain. There may be somewhere between 5 and 10, so not that many, but these structures are quite interesting. If we do now a close-up of this sort of staining, in the center of these, what we now call fungal-induced glial granulomas, or FIGs, you see these very abnormal-looking nuclei, which then if you do a calcifer white stain, which picks up the chitin, which is the polysaccharide unique to fungi, you realize now that this is a bunch of fungi that have been herded somehow into these spherical structures. And again, we see the activated microglia and astrocytes. A little bit more staining now, looking for A beta. Here we're looking at one of these fungal-induced glial granulomas. This is about a quarter of that FIG. And we can see that A beta now in red is now seen. It's completely decorating those fungi. So somehow that A beta is being carved up from the amyloid precursor protein and then basically being thrown on top of these fungi that are at the center of these FIGs. In the past, it was not really known so much if the mouse could actually produce A beta peptides. That's why, for example, human A beta or amyloid precursor protein transgenic mice were developed. These data clearly show that you can make full-length, in additional data, full-length A beta peptides in the mouse, strictly from their own endogenous amyloid precursor protein. A little bit more about this infection in terms of its duration. Here we're looking at cultures of mouse brain. So here we're looking at 2 days post-infection, colony-forming units per gram of mouse brain after the same injection of 25,000 organisms intravenously. So looking at different ages of mice, 6 weeks, 38 weeks, and 53 weeks at the time of infection. You can see against a few hundred residual fungus after 2 days, but then up to 10 times more fungi in these aged mice, indicating that age is a very important result in terms of how efficiently the mouse can clear out this organism from the brain. Looking more at a kinetic experiment, so comparing fungal burdens in the brain at 2 and 10 days after infection. Again, 6-week-old mice are very efficient. By just 10 days, that organism is almost completely cleared from the brain, but quite a bit of residual fungus and even more beginning at 2 days. So less efficient clearance of this organism from the brain, again, in an age-dependent manner. So in summary, to not have to torture you with a whole bunch of primary data, what we've shown is an intravenous injection of Candida albicans leads to this very interesting infection involving the formation of fungal-induced glial granulomas that have this characteristic pattern with fungus in the center, surrounded by an increased expression of amyloid precursor protein and decorating the fungi themselves are A beta peptides. At the same time, this FIG is being formed. The mouse brain is secreting the inflammatory cytokines have already been linked to Alzheimer's disease, for example, tumor necrosis factor, IL-1 beta, IL-6, and type 1 interferons. You get activation of these microglia. They're clearly able to kill this organism over about 10 days in young mice. But during that time that they're killing these mice do show impaired spatial and working memory, many features, again, of Alzheimer's disease. We wanted to know what is the clearance mechanism for this organism. So let me just show you a little bit more data here before I turn it over to Lynn. We wanted to know where's that A beta coming from. So here in this experiment, we're taking the secreted aspartic proteases, or SAPs, of Candida albicans, provided by one of our collaborators, Bernie Huber, in Jena, Germany. And then we simply added that to cultured into A cells, which are transformed neurons that express amyloid precursor protein. And by ELISA technology, we can readily detect A beta 40 and 42 after just 24 hours of culture, but we can do a much simpler experiment. We can simply take recombinant amyloid precursor protein and mix the same combination of Candida albicans secreted aspartic proteases. We get exactly the same thing at different pHs, including the optimum pH for these aspartic class proteases. The importance of these experiments, so it's well known, of course, that endogenous secretases, which are also aspartic class proteases, are important in generating A beta peptides endogenously. Here, what we're showing for the first time is an absolutely exogenous pathway for generation of A beta peptides that is organism dependent. And these are from the SAPs of Candida albicans. And then just a Western blot, here's A beta. After culture, we see now peptide fragments that react to an A beta specific peptides of approximately the right molecular size. So these indeed are A beta-like peptides. A little bit more about the immunology. So we wanted to dig into what are the mechanisms that, again, that lead to the resolution of this infection. So here we're showing you a bit of a clearance experiment in which we're injecting the same Candida albicans. Here's our wild type mouse that has, roughly 300 to 400 organisms after 4 days in the brain, and that is cleared out virtually completely at day 10. But if we look at mice that are missing amyloid precursor protein or they're missing toll-like receptor 4, I'll explain in a minute why we did this pairing, then what we see is pretty much identical clearance curves of these two gene knockout mice, but clearly they are impaired in their ability and less efficiently able to clear this organism. But ultimately at day 10, everyone's able to clear this infection out. So A beta, APP, TLR4 seem to be important, but they're not as absolutely essential because ultimately everybody can clear this infection. So something else is going on here. We wanted to track that down. So we wanted to focus on other than SAPs. We wanted to look at other virulence factors from Candida albicans. And so we wanted to look at this guy here, Candida lysine, which is a product of the ECE1 gene of Candida albicans. This is a peptide toxin secreted by Candida albicans, was discovered by Julian Naglik, our collaborator in 2016. This is a cytolytic major immune activator. We wondered if it might be important in cerebritis. And again, in the interest of time, I won't torture you with primary data. Let me just summarize this in the context of some of our other work that we have published more recently. So once we inject Candida albicans intravenously, once it gets into the microcirculation of the blood-brain barrier, Candida albicans adheres to the endothelial cells and begins secreting those same secreted aspartic proteases that are crucial to dissolving the tight junction proteins that are essential for getting the organism into the brain, at which point it forms these fungal-induced glial granulomas. And then we've discovered a couple of different clearance mechanisms that are activated through the various virulence factors of this organism. First of all, again, as I mentioned, SAPs will cleave amyloid precursor protein into A beta peptides. These then act as ligands for toll-like receptor 4 that activates microglia to secrete antimicrobial peptides, make them more phagocytic and able to intracellularly uptake this organism for intracellular destruction. But there's an even more important mechanism that's through that Candida lysine I just briefly introduced you to. We discovered the second mammalian receptor for Candida lysine, which is the integrin complex MAC1 or CD18, CD11B. Candida lysine engaging MAC1 and CD11B specifically further activates microglia to secrete those antimicrobial peptides and lead to enhanced killing of this organism. Okay, so that's what we have with our intravenous injection model. We have a much better model now, and to tell you about that is an exceptional postdoc in the laboratory, Lynn Bimler.
Thank you, Dr. Corry. And now I get to torture you with a little bit of preliminary data. So this is what we proposed as our initial chronic model for cerebral mycosis, because while our IV model was showing some really promising results, the main issue is, first of all, it's not necessarily translational, and Alzheimer's is a chronic issue. It's not something that can be resolved in 10 days. So we wanted to come up with a solution that would be something that is not only translational and more similar to the human condition, but also is something that can take place over a long period of time, possibly the lifetime of the mouse, to see if we developed kind of a true dementia phenotype. And so our initial hypothesis was that we could give an oral gavage of Candida albicans to establish a colonization of the GI tract similar to humans, and then a transfer would occur of Candida albicans into the bloodstream, which would result in a brain infiltration of Candida albicans into the brain, similar to what we just showed in the model Dr. Corry described, and this would eventually lead to dementia. So the first question is, does this happen? And the answer is yes. So we can establish a colonization of the GI. Here we're looking at the fecal matter within the lumen of the intestine. And we see consistently out to 6 months, and we've even shown out to 9 months, we have colonization of the GI. We also can see that the brains of these mice have elevated infection going out to at least 6 months. Compared to controls. And we do get some baseline level of infection in brains in mice, even when we take them straight from our vendor source. So we're really interested in the statistical increase in infection. When we look at the brains of these mice, we pull up the yeast inoculum that we are infecting them with by oral gavage, or we can compare to the brains, the colonies taken out of the brains from the IV model. And these look like classic yeast colonies. You can see how the colonies look on the plate in the bottom of the screen. And then you can also see that we have some nice, beautiful looking yeast cells with a small amount of filamentous cells. But when we took out the brains from the oral gavaged mice, we found that these are morphologically irregular. So they don't look like traditional Candida colonies. Some of them even have kind of yellow patches. They're amorphous. They were not what we were expecting. But when we look under the microscope, we can see that there's a lot of things going on here. There's a sea of bacteria, so we have these smaller cells, but then we also have larger yeast cells, some of which are even budding, and we have pseudohyphal and filamentous cells as well. And so this is actually a polymicrobial biofilm that's growing out of the brains of these mice. And sometimes we even see these cells that look like yeast cells attached to bacterial cells. So our chronic model now takes a little bit of a different shape, because during this GI colonization, we actually have a interaction with bacteria that leads to polymicrobial units that are theoretically leaving the gut and metastasizing to the brain and then causing this chronic infection. And so we found that FIGs are also present in the brains of these mice. So here you can see that we have calcifer white staining as well as IBA1 and A beta staining in localized regions, similar to what I just showed you we just showed you in the IV model. We see low numbers of FIGs in these mice consistently going out to 6 months. And the FIGs are generally in regions that are often affected by Alzheimer's disease, including the cortex and corpus callosum, hippocampus, and hypothalamus. We also see that these regions, or many of these regions, have decreased brain volume by 6 months. So when we do an MRI of these mice, we see that the pons medulla, the thalamus, hypothalamus, and superior colliculus, and the cortex all have decreased brain volume. We also see we almost have a significant increase in, or we have a trend towards an increase in ventricle size. And all of these are linked to AD in humans. Finally, we did some work doing long-term potentiation with a collaborator to see if the actual brain function is inhibited. And we found that actually there's almost a complete abrogation of the brain function of the hippocampus. And I'm not sure why it's not showing up here. There should be lines here for these two groups. This is really tragic because this data is beautiful, but this 100 is basically the baseline. And these two groups, their trend line is baseline. There is no function coming out of the hippocampus. And finally, we also wanted to just actually check the behavior of these mice. So we found that if you put them in a novel place, most mice will groom. So this is a novelty-induced grooming test. But these mice actually jump and try to escape. So if you count those jumps, you can see how anxious the mouse is. And as you can see, these mice are significantly more anxious than controls at 2, 4, and 6 months. And some of these mice, there's actually a little plastic lid, and they're bumping their head into this lid 20 times sometimes. So these are extremely anxious mice. It would take a lot for me to do that. And so we did a number of tests, but we also did a conditional fear test, which shows a more significant dementia. So these mice are placed in, a novel environment. They're allowed to explore, and then the floor is electrified, and it's paired with a tone. The next day, we put these mice back into this environment. And again, we see that these mice don't appear to remember that they've been shocked. A normal mouse will freeze, but our mice freeze less than the normal mouse, which this is a pretty severe learning inhibition. And so finally, Does this translate to humans? The colonies from the human brains are actually, that we have cultured, are actually also polymicrobial. Here you can see that we have yeast cells, some of which are budding, as well as pseudohyphal and filamentous cells in a sea of bacteria, similar to what we saw in the mice. And again, we sometimes have the larger yeast cells with bacterial cells. And we've also done this through scanning electron microscopy, where you can see the bacterial cells and the fungi on a matrix of what looks like filamentous fungi. And through sequencing, we again have confirmed these identities. So our conclusions are that SAPs drive fungal entry into the blood-brain barrier, and we have evolved robust mechanisms to counter this through SAPs and A-beta Toll-4 axis, Candida lysine and MAC1 axis, and Candida albicans is the first microorganism identified that can alone cleave A-beta or APP into A-beta peptides. Candida albicans gut colonization persists for the lifetime of the mouse, and this is likely a persistent source of polymicrobial brain infection acquired metastatically. Finally, there's also a bacterial component deriving from the gut. So overall, a persistent, potentially lifelong polymicrobial cerebritis of the mice causes devastating neurophysiologic impairment that manifests as increased anxiety, memory loss, retinopathy, which we didn't show here today, and complete loss of hippocampal long-term potentiation. The human AD, but also non-AD brain contains similar Candida albicans-based polymicrobial affiliations. And so we posit that Candida albicans is a candidate for a microbial cause of degenerate brain diseases related to polymicrobial cerebritis, including Alzheimer's disease. And so we'd like to thank our funding sources, and we're open to any questions if we have time. Thank you.
Lynn, David, thank you so much. Thank you to everyone who presented the first session. We do have some questions that came in from the chat, and I'll start with one specifically for Lynn and David. The panelists wanted to know, or one of the people joining us online, have you looked at the CSF of the mice? And if so, have you seen cultures of the CSF to see if you can grow microorganisms?
We have not yet looked at the CSF, but
I don't think we know how to collect mouse CSF, but it's a good thought.
we can learn.
Thank you. Are there questions from the audience? When I see you. Yeah, you can speak into, if you could talk into the mic for the online folks,
Okay.
that's fantastic. Thanks.
So, besides the oral gavage, the gut, oral candidiasis is a big problem, very popular. Actually, when you have HIV, the first one to show up is actually the oral candidiasis. And you got the blood, the main blood supplied, to the brain is actually through oral. And I was wondering, there are a lot of good models there for creating an oral candidiasis, and that could be something.
So that, yeah, I think it's a great point. So yes, Candida can be found all over the body. It could be in the skin, it could be in the eyes, the oral cavity, esophagus, stomach, throughout the GI tract. Yeah, so it could be coming from many different sources. We focus on the gut because of the polymicrobial nature, the bacterial signal, we didn't have time to go into, but the bacterial signal is coming from the gut. It's not coming from the mouth, it's coming from the gut. But as we work through our human data, which we don't have quite organized just yet, but we're going to be, it's going to be very instructive to understand what are the bacteria affiliating with Candida from human brain. So if that's Porphyromonas gingivalis, well, yeah, it's probably coming from the oral cavity. If it's Escherichia coli, yeah, it's coming from the gut, right? But maybe we're going to get a mix. I don't know. So we'll just have to see how that goes.
And we have seen at least some what looks like infection in the sinus.
Yeah.
It's possible we're also in the mouse model. It's possible we're also getting some in the mouth because oral gavage is going through the mouth. So it's possible that this model is actually potentially showing results of that, even though we're focused on the GI, and that's where we're finding a lot of the Candida because that's where we're looking.
Yes.
Corey, I'm so sorry. Can you turn your mic on? I was running around turning those
That's,
on.
I thought I was going deaf. Yeah.
Now, can you hear me now?
Yes.
Better.
Okay, great. The phenotypic data, I think, is really cool how the lack of, or the decreased volume and the lack of potentiation in the hippocampus. So I'm curious, do you think that neuronal loss is happening with this? And if so, what do you think might be driving that?
So I think the question was, are we seeing neuronal loss at the same time we're seeing brain shrinkage? So we haven't done quantitative pathology to really answer that question, although we have our collaborators who we're working with on that. We're planning that for the next stage of experiments, but we suspect that yes, loss of brain size is at least in part due to loss of neurons, but we have not proven that yet. But Lynn,
We do have decreased synapses. So we haven't
that's right.
done any staining for if there is neuronal death, but we do have staining in both the retina and the brain showing that you have decreased synapses. And initially there was some data that maybe in the retina there was decreased decreased neurons, but I don't think it panned out. So
So
nothing yet.
And in terms of mechanism, what might be driving, is there anything you understand about albicans? I mean, there was the peptide, I think that was neurotoxic or something, you made the major one, but what might be driving that, or what's your best guess?
yeah, so that's a really good question. We don't know, have any insight into what might be causing neuronal loss. One thing to point out though is that what's already been established is it's type 1 interferons that are produced in the Alzheimer's brain, but also in our model. And that's already known to be neurotoxic. So we kind of already have that. There's already one part of that mechanism sort of already in place. But it's not really clear in my mind anyway why type 1 interferons would cause synaptic dendritic pruning and synaptic loss and that kind of thing. But at least that it's already known that that is more or less a neurotoxic cytokine milieu that we already have. But there's probably much more to that. So that's some of the future work.
Thank you.
Yes, in the back. So yeah, the question is do we have a correlation between the duration of the infection, which is again, as Lynn pointed out, is lifelong in the mouse, and severity. And so yes, Lynn, can you maybe talk about the long duration experiments you've been tortured with?
So some of the 6-month data, it looks like over time, the mice behavioral phenotypes get worse. The LTP, honestly, it looks very similar. It basically immediately after infection, we think it abrogates a lot of the brain function there, which should inhibit learning. We have also looked at we've only looked at 6 months for the MRI and brain shrinkage, but presumably there's no shrinkage before they're infected, so it's getting worse theoretically. And then I don't think we've looked at the neuronal synapses at multiple time points. We've just looked at 6 months as well. But the behavioral phenotypes do get worse, except for the conditional fear, which we've been told by behavioral experts, the reason we might lose significance there at 6 months is just because mice stop just through natural aging doing as well in that test. So they said the fact that we even have a trend at 6 months when the mice are about 9 months old is pretty impressive.
One more quick question.
Sure.
So you're saying that fungus gets into the brain and it slowly turns us into zombies like that TV show?
Mhm.
That's not the real question, but
Yes.
I'm gonna miss this. Is the fungal, is it all throughout the brain or there's certain areas where it prefers to colonize?
Great.
And Michiko, this is a question for you as well, as far as where the bacteria is.
Yeah, so the question is where basically geographically is that fungus, and these polymicrobial infections going? So we were actually still quantifying this, but so far our data indicate it's rather diffuse. And so every region that we've looked at so far, although again, those studies are still going on, correct me if I'm wrong, Lynn, but we found it just about everywhere.
So I think in the IV model, it was a little more diffuse. And then in the oral gavage model, I think we pretty consistently out to 6 months found it mostly in the cortex and corpus callosum, the hippocampus, and the hypothalamus. But this is also, the way they're testing for these is through serial sections. So they're doing maybe 10 serial sections in one brain. So it's also possible that they're in other regions and that it's just a, sampling error.
So for your answer, we actually just experimented only 30 subject of brain, so we haven't really able to conclude if it shows up like extremal on frontal or temporal and horny cortex, just those 3 locations right now. Once we have more brain region, we'll be able to test more. And we just got CSF and also primary motor cortex from University of Wisconsin, so I'll be able to answer more in the future.
Just one more quick question, so we have time
Yeah,
for a brief bio break.
I have 2 questions, but I
Go ahead,
will just
Lavinia.
reduce it to one. I have a question to Garth and Michiko. Can you do single cell pack sequencing? Because there are intracellular bacteria microbes and extracellular, and somehow with a differential sequencing from the whole sequencing to single cell, as your sequencing pack bio capabilities are very, very accurate, probably can give us a little bit more insight about what is intracellular, what's extra.
So the question was, can we use our assays to look, at single cells as opposed to, mixed cells? So yes, we could. What Michiko's actually been doing has been looking at what we call, heterogeneity within it. So she has a particular specimen, brain specimen, that's 1 centimeter or something like that, cubed. And so she'll take multiple punches from within that. And we actually see variation in terms of, the relative numbers, of the species, even within, the different punches. So we know that there's heterogeneity, even at that kind of macroscopic level. And so one of our plans is obviously, to get smaller and smaller. We're working with a frozen specimen. It might be very difficult to get a single cell, just because of the mechanics of it. But there's certainly no reason why we can't, if we were working with somebody who was comfortable in isolating single cells, we of course can use our assay on any specimen type. So if there are people who can isolate, single cells from frozen brain tissue, we'd love to work with you.
And I just, one more question regarding the clearance to David and Lynn. So the clearance, does it happen through the same mechanism? Is it through the lymphatic system? Is it through the vasculature? How does it happen? The clearance of Candida.
So, thanks. Correct me if I'm wrong. The question is, how is clearance of the organism accomplished? And so, what our work is focused on really is through the microglia. So we think that it is a combination of, first of all, increased phagocytic uptake and intracellular destruction and elimination on that basis, but also release of antimicrobial peptides in particular that lead to extracellular killing and then cleanup of the debris. There probably are other mechanisms, but we have not looked at that yet. I hope that answers your question.
Yeah, thank you.
Also see activated astroglia, but we haven't looked at how that clears or anything. So we know there are probably other mechanisms. Thank you.
I have one last question. All right. So Garth, your method that was using PacBio is very interesting. I'm curious as compared with deep sequencing, in terms of the sensitivity. I know you're reading longer, but in terms of the sensitivity and also at the price point of view, how are you going to compare that, make it a
Because we're starting out as amplification based. We have essentially limit detection. We've demonstrated with reconstruction experiments that we can detect for most bacteria, a single organism, obviously, you're always dealing with a Poisson type of distribution, but our sensitivity is extremely high. Now, if you've got organisms that are present, at 6 orders of magnitude more than another organism, the only way you get around that is by, sequencing extremely deeply. But probably, although you never know for sure, if an organism is representing less than 1 part in a million, it's kind of our assumption that it may not be particularly important, which may be wrong, But as I said, we showed that we are linear over about, 4 orders of magnitude, with our assay. In terms of cost, we can multiplex on the PacBio system, between 100 and 150 specimens on a single SMRT Cell. So it's not ridiculously expensive. It costs, in terms of like, it's around $100, per assay per specimen.
Great, I want to thank all the speakers for this morning as well. And we'll take about a 5 to 6 or 7 minute bio break and then reconvene, okay?